Let's discuss lysosomal storage disease. And before going dive into the lysosomal diseases, let's elaborate briefly the general cycle of lysosomal storage enzymes, lysosomal enzyme, how they interrelate, how they convert from one form to another form, and how this whole cycle is being happen in the body. So starting with the ganglioside 2. This ganglioside is converted by an enzyme uh, hexoaminidase enzyme. This hexoaminidase enzyme, it converts GM2 ganglioside into GM3, which directly convert into glucocerebroside. This glucocerebroside is then converted by an enzyme known as glucocerebrosidase enzyme. So I just wrote it like this: glucocerebrosidase enzyme, and it is converted into ceramide, the final product of lysosomal enzyme. So this ceramide is the main component in the lysosomal substrate conversion. We have another substrate known as sphingomyelin, and if this sphingomyelin is broken down, then it is converted into ceramide. So this whole process is like pretty much straightforward. So GM2 is converted into GM3, GM3 readily converted into glucocerebroside, and which directly broken down to form ceramide. Another method to produce ceramide is sphingomyelin. We break the sphingomyelin by sphingomyelinase enzyme, and it gives ceramide. Another pathway, there are two other pathways which helps us to give this glucocerebroside. So the first one is globotriacyl ceramide or ceramide trihexoside. So I just wrote it like this: globotriacyl ceramide. Globotriacyl ceramide is one name, or it is also known as ceramide trihexoside. So I just combined two names to just make a visual memory that this is a same component. So rather learn the two different names, we just combine it for our convenience. So globotriacyl ceramide trihexosidase is converted by an enzyme known as galactosidase, and this enzyme gave us the product glucocerebroside. Similarly, we have another substrate, cerebrosulfates or sulfatides. So I just wrote it like this: cerebroside sulfates or sulfatides, just for my convenience, and we convert it by, this is a sulfate, so we need a sulfatase enzyme. So there is an enzyme known as aryl sulfatase enzyme. It converts this this cerebroside sulfatide and give us galactocerebroside. This galactocerebroside is now converted by enzyme similarly by an enzyme like glucocerebroside, and we have an enzyme known as galactocerebrosidase enzyme, and it gives ceramide. So now we have 1, 2, 3, 4, 5, 6 enzymes we have. So this is the whole diagram, whole chart of whole algorithm regarding the uh, lysosomal storage diseases. So we just track this whole pathway and follow the different problems. So if there is enzyme deficiency, I have wrote this whole topic on this slide. So if we have the enzyme deficiency of hexoaminidase, so no hexoaminidase, no GM3, no glucocerebroside, no ceramide, so we have accumulation of GM2. So no hexoaminidase, we have increased level of GM2 gangliosides, and which give us these clinical symptoms. So why I wrote Tay Sachs disease and Niemann-Pick disease in a tabulated manner, because their presentation are pretty much similar, and there is a pretty much chance that we just confuse Niemann-Pick and Tay Sachs disease, because there is two symptoms, neurological decline and cherry red spot, which are present in both of the disease. So the differentiating presentation is hepatosplenomegaly. So let me break down this. We have GM2 hexoaminidase deficiency, or we have sphingomyelinase deficiency. If sphingomyelinase is deficient, then obviously we have increased level of sphingomyelin, which is known as Niemann-Pick disease. So in Niemann-Pick disease, we have no sphingomyelinase, and in Tay Sachs disease, we have no hexoaminidase. So if both are absent, and we have increased level of GM2 and increased level of sphingomyelin. So these GM2s and sphingomyelins, they are very much neurotoxic. They can cause progressive neurological degeneration, along with cherry red spot. Because eye is a derivative of our brain tissue. So if the brain is damaged, then relatively the retina, the eye is also damaged. So the neurological deficits, the presentation is pretty much similar. So the differentiating factors between two problems is hepatosplenomegaly. So we have hepatosplenomegaly in the Niemann-Pick disease, but we don't have hepatosplenomegaly or altered ALTs or ASTs in Tay Sachs disease. So these are the differentiating factors. In Niemann-Pick disease, we have also foam cells. With foam cells are the lipid-laden macrophages. This is pretty much a short description of presentation of Niemann-Pick. So the Niemann-Pick, we just keep pondering on the hepatosplenomegaly. In the question scenario, if we see cherry red spot, if we see progressive neurological decline, then we have to find if there is altered ALTs or ASTs, if there is cirrhosis, if there is hepatosplenomegaly, if there is any hepatosplenomegaly related problem, anorexia, abdominal pain, whatsoever, then it is definitely Niemann-Pick without any doubt. But if there is no such thing, then we have to rule out Niemann-Pick disease and go for Tay Sachs disease. In Tay Sachs disease, there are also other presentations, especially the lysosomal cell, onion skin lysosomal cell, There is abnormal hyperreflexia, or in hyperreflexia, if there is a startle reflex, which is also a reflex, so there is abnormal startle reflex because we have hyperreflexia. So neurological defects with hyperreflexia, abnormal startle, hyperacusis, they all go for Tay Sachs disease. The next problem is Fabry disease. Fabry disease is due to the deficiency of galactosidase. So if we track this one, globotriacyl ceramide or ceramide trihexoside is converted into glucocerebroside substrate. So if they by the help of galactosidase, if this is deficient, then we have increased level of globotriacyl ceramide trihexoside. We have increased level of globotriacyl ceramide. So no galactosidase, increased level of globotriacyl ceramide or ceramide trihexoside, and these lead to triad or episodic. There are two different types of presentation, early and late. In early presentation, they are episodic peripheral neuropathies, skin problem, and sweat hair problem. So they have hypohidrosis, like connective tissue or skin related problem, and there is neurological problem. So we have peripheral neuropathies along with angiokeratoma or hypohidrosis, and they are present intermittently or episodically. Similarly, if the patient is untreated, then the complications are the renal failure and cardiovascular problem, including stroke and left ventricular hypertrophy. Then we have Gaucher disease, and in the Gaucher we have no glucocerebroside enzyme. So we have this their problem, we have the problem in this conversion. So glucocerebroside is converted into ceramide. So if this glucocerebroside enzyme is deficient, we cannot make ceramide. So we have increased level of glucocerebroside. If this happen, then it can lead to hepatosplenomegaly and pancytopenia. Also, it is related to osteoporosis and avascular necrosis of the femoral head. There is also a clue in the correct question scenario that the tissue crumpled macrophages are present on light microscopy. So tissue crumpled is again go for Gaucher disease. We have discussed AVN or avascular necrosis. The etiology of avascular necrosis is Gaucher disease. In osteoporosis, the etiology of the patient has Gaucher disease. So pancytopenia and hepatosplenomegaly are the most important clinical scenario. Rest of them are the associations. The next one is the low yield two problems, metachromatic leukodystrophy, and it is due to the absence of aryl sulfatides or decreased activity of aryl cells. So sulfatides are converted into galactocerebroside. So if this enzyme is deficient, then we have increased level of sulfatides or increased level of cerebroside sulfates. So no aryl sulfatase, increased cerebroside sulfates or sulfatides. Now these are very much demyelinating agents. So they demyelinate whole neurological system, including CNS and PNS. And this demyelination in the CNS is lead to dementia and ataxia. Then we have the Krabbe disease, and in the Krabbe disease, if we have the deficiency of our last enzyme galactocerebroside, then we cannot make ceramide. We have increased level of galactocerebroside. So no galactocerebroside, increased galactocerebroside, which can cause oligodendrocyte to destroy and if oligodendrocytes are destroyed, we can lead to a problem known as optic atrophy and peripheral neuropathy. And in this Krabbe disease, we have the globoid cell. So these are pretty much low yield 2 problems. The next one is Hurler and Hunter disease, and they are pretty much related to mucopolysaccharidosis. So in the mucopolysaccharidosis, we have Hunter and Hurler disease, and in these diseases we have the similar conditions, but we have two differentiating factors. First one is we have no L-iduronidase enzyme, which broke the iduronate in substrate and make heparan sulfate, which broke the heparan sulfate and make iduronate enzyme substrate product. So no iduronidase enzyme, then increased level of heparan sulfates. And if we have no iduronate 2 sulfatase enzyme, then we have the Hunter disease. So in the Hurler disease, the L goes for the L-iduronidase enzyme, and the Hunter, the T is go for iduronate 2 sulfatase enzyme. All are T's. So if we have deficient these enzyme, then we have increased level of dermatan sulfate or heparan sulfate. We have hepatosplenomegaly. We have developmental delay, neurological decline. We have airway obstruction, skeletal abnormalities, especially the face abnormalities. So we have neuro decline, face abnormality, and increased level of heparan sulfate. So there is one differentiating factor, which is corneal clouding. So in the Hurler syndrome, we have that corneal clouding or cataract like condition, but in the Hunter problem we don't have that much problem. So Hunters are very much aggressive. They are very much targeted for their prey. So if a person is very much targeted, then it doesn't have any eye problem. So Hunters have no eye problem, Hurlers have eye problems. So these are all about lysosomal storage disease.